Here we are calibrating the pH meter using a two-point method. First, we rinse the electrode with distilled water and place the electrode in one of the buffer solutions. We then wait for the voltage to steady out and then record that pH. We then rinse it again and move on to the next buffer. After you record that pH, your meter is now calibrated. Here we are using the pH meter to determine the pH of orange juice by rinsing the electrode and then immersing it in the orange juice. We wait for the pH to steady out and then record the value in our notebook. We then properly rinse and store the electrode for later use. Next, we use the pH meter to determine the pH of apple juice by rinsing the electrode and then immersing it in the apple juice. We wait for the pH to steady out, and then we record the value in our notebook. We then properly rinse and store the electrode for later use. Next, we use the pH meter to determine the pH of hydrochloric acid, which has a concentration of 0.20 molarity. We first rinse the electrode, then immerse it in the hydrochloric acid. Then, we wait for the pH to steady out and record the value in our notebook. Finally, we use the pH meter to determine the pH of ammonia, which has a concentration of 0.25 molarity. We first rinse the electrode, then immerse it in the ammonia. Then, we wait for the pH to steady out and record the value in our notebook. We then properly rinse and store the electrode for later use. For a monoprotic acid HA with conjugate base A, the equilibrium constant Ka is represented by this equation. Ka, the acid dissociation constant, quantifies the extent of dissociation. The larger the value of Ka, the stronger the acid. The smaller the value, the weaker the acid. Strong acids dissociate completely into their ions in water. The dissociation of a strong acid is represented with a one-way arrow, as its equilibrium lies far to the right. On the other hand, weak acids only partially dissociate. Because of this partial dissociation, both the acid and its dissociation products will be present in the aqueous solution and will react to equilibrium. This is represented by a double-ended arrow. A buffer is a solution that resists pH changes. It is created from a weak acid and its salt, or a weak base and its salt. Titrating a weak acid with sodium hydroxide produces its conjugate base pair that prevents the pH of a solution from changing. When a strong base, such as sodium hydroxide, is added to the buffer, the acetic acid reacts with and consumes the excess hydroxide ion. The hydroxide reacts with the hydronium ion from the acid in the following reaction. Hydroxide reduces the hydronium concentration in the solution, thus preventing pH changes, and shifts the equilibrium to the right.